Okay, welcome to our final installment in the trilogy of the modes of inheritance. Today we're going to be looking at the codominant inheritance of alleles. By the end of this lesson, you should be able to, number one, define what codominant inheritance is, and then demonstrate that understanding of inheritance using Punnett squares. A little review on the three types of inheritance of alleles. The first type of inheritance was our complete dominance of one allele over the recessive allele. So in the complete dominance of inheritance, here we are looking at the complete dominance here. We have a capital letter representing the dominant allele and a lowercase letter representing the recessive allele. And then we have an inheritance where if the heterozygote exists, the dominant allele is always manifested in the phenotype or the characteristic trait, not the recessive allele. Last lesson we looked at incomplete dominance of alleles. So here we had the heterozygote where the F1 generation, they were to receive one or both alleles, both different alleles. If they were a heterozygote offspring, then both alleles were expressed. There was no dominance of one over the other, although the heterozygote offspring showed a distinct phenotype different to both parents. In codominance, the last type of inheritance, we have alleles, some of the alleles who are codominant, meaning they both are expressed at the same time. Just like the incomplete dominant, the only difference here is with the heterozygote offspring, we have two different alleles, it being expressed, and they both have similar characteristics to the parents. Okay, a really common example of codominant inheritance is the four main blood types in humans, which we know, of course, are blood groups A, B, AB, and O. So the gene that determines blood type has three different alleles, which code for a protein we call immunoglobulin. These alleles are capital I, and that stands for immunoglobulin, capital I with a superscript of A, so that's our blood type A. Then we have immunoglobulin B, and then we have our underscore or our lowercase immunoglobulin. Note, I just wanted to bring you over to this note here in the blue. The allele I, the lowercase I, which represents blood group O, is recessive to both of the immunoglobulin A and immunoglobulin B. Now, immunoglobulin A and B are both codominant to each other, which means when an individual has both of those alleles, then both of the immunoglobulin proteins are going to be expressed in the heterozygote. Okay, all individuals inherit one allele from each of their parents. And so, of course, the combinations of alleles could be any of these, what have we got? 7 different combinations of alleles here. So we have immunoglobulin A, one allele from mum, one allele from dad, and of course they're codominant with each other. So they're both going to express that same blood type protein, immunoglobulin protein. These are homozygous. And then we have immunoglobulin A, one from mum, and another one from dad, the recessive. So this immunoglobulin O is not going to be produced because it's recessive to the immunoglobulin A protein. So our blood type is going to be A in this case, but because we have two different alleles, it's going to be a heterozygote. And all the way down, so we have immunoglobulin B twice over. This is a homozygous offspring, and so their blood type is going to be B. And then because the recessive allele here for O is recessive to immunoglobulin B, we have the blood type being a B blood type, heterozygous. We can go down here, immunoglobulin A from mum or dad, and the other one from the other parent. And so, of course, because immunoglobulin A and B are both codominant to each other, we have two proteins being produced, immunoglobulin A and immunoglobulin B, and the individual is blood type AB. Now we can switch those around. It could be B from mum or B from dad, or A from mum or A from dad. It doesn't matter which way you have them around. They're both going to be heterozygous for the blood type AB. And then finally we have our two recessive alleles, I, lowercase I and lowercase I, resulting in a blood type O. Okay, so we've got a nice diagram up the top here. Now this immunoglobulin protein that's produced from these alleles, this gene, is a protein marker which is carried on the surface of red blood cells. So you can see that they have different shapes, and so type A looks like this with the blue ball, type B has the purple arrow, and then type AB has both of those proteins being expressed. Now genotype lowercase I, blood type O, results in no protein being produced at all, and so these type O blood cells don't have any of these immunoglobulin markers on them. Okay. All right, so here we have our codominance. So in this example, we're looking at codominant inheritance, not the recessive versus a dominant, but we're just going to be looking at these two codominant alleles in this example. So in codominance, when both parents are homozygous, the offspring will be heterozygous and its phenotype will display the phenotype of both parents. So if we have one parent type A, and they have to be homozygous for that type A, so they've got to be immunoglobulin A from their mother or father, immunoglobulin A from their mother or father, so that's one individual. And then the other parent is homozygous for immunoglobulin B, type B, so they're going to be of this blood type. Okay, so if these two people get together and reproduce, they have a child, what are the probability that their children are going to be either type A, type B, or type AB, or even type O? So let's do the Punnett square cross, and we can drag and drop this one down and down to here and here, and then we're going to move those across, IB and IB, IB and IB. So you can see that all 25, 25, 25 and 25% will all be heterozygous genotypes for blood type AB. They're going to be the heterozygous for both immunoglobulin A and B. Another example for codominant inheritance in humans, we have sickle cell, which is a mutation to the shape of the red blood cell. It affects the shape of the hemoglobin, and therefore the hemoglobin affects the shape of the red blood cell, and it also hardens that red blood cell as well. So it reduces the amount of oxygen carrying hemoglobin in the cell, affects the shape, of course. Now we know that the typical biconcave shape of the red blood cell is quite flexible, and it gives a large surface area to volume ratio, making it ideal for oxygen to diffuse in and out of the cell towards the needed cells that require oxygen. Whereas individuals who have sickle cell, they have these hardened versions of cells with lower surface area to volume ratio, and they're quite jagged, and they tend to clump in the bloodstream and cause blockages in small capillaries and veins, and causing issues with those individuals' health. So here we have one homozygous normal red blood cell parent, and then we have an individual crossing with them who have homozygous SS for sickle cell. So they have two alleles for sickle cell gene. When these two homozygous people cross, they produce an offspring. Looking at our Punnett square, 100% chance of that offspring having an intermediate variety of the sickle cell. Okay, so we have some of the normal cell red blood cells being produced and some of these sickle cells being produced as well. Our last example is our roan cows. So we have two different colouring cows, and the gene that expresses these colours we write the genotype as capital C with a superscript R, so that C stands for colour and the R stands for red, colour red, so 2 alleles that mark for red colouring, and then we have 2 colour alleles standing for colour white in the other homozygous parent. When these two cross, both alleles are expressed in the heterozygote offspring, but you can see distinct colourings from both parents. So in the heterozygous offspring, both parents' alleles are expressed in the phenotype. Okay, and we can evidence that using our parental cross here. In the case of the F1 offspring here being our colour roan, so it has those 2 heterozygous alleles, colour red and colour white, and we have that speckled version. If we were to take 2 of these F1 offsprings and then crossmate those, we would then have a more of an array of the different phenotypes in the F2 generation. So we can see 1 red allele going to this cow from the parent 1, and then the other red allele going from parent 2, creating that homozygous colour red, just like the parent 1 up here. Okay, and then we have our intermediate roan cows again, 1 red allele and 1 white allele from the parents, and again 1 red and 1 white allele. And lastly, we have our homozygous white coloured cow with a white allele cross.